Now we're going to look together at our notes number 2, evidence of evolution. Scientists use many kinds of data to piece together the evolutionary history of organisms. This data comes from fossils. We look at fossil records and comparative anatomy, compare how maybe the arms of one organism match up with the arms or wings of another organism. Embryology, remember, embryo is when we get an egg and a sperm. The sperm fertilizes the egg, we get what's called a zygote, and then after it goes through mitosis a few times, we call it an embryo. So embryologists study that early development of infants. And then we look at the biochemistry. We now have technology where we can go in and look at the actual chemistry that makes up the different organisms, like archaeobacteria, eubacteria. They used to be categorized just as Monera single-celled organisms. But now we have the biochemistry technology to be able to go in and see that there are actual chemical differences in the cell membrane, or the cell wall of the bacteria and the eubacteria. Some have peptidoglycan in them, some do not. So now we can change classification based on the fact that we can even go in and look at the actual chemicals that make up the parts of those cells. Fossil evidence of evolution. Fossils are the remains or the traces of organisms that lived in the past. Scientists use relative dating to estimate the age of fossils, such as carbon-14 radioactive dating, or relative dating based on, they'll look at the way the rocks are stacked and can estimate which rocks came before the other rocks, so which fossils in those rock layers came before. There are things that can change the way that they're arranged, though, so the relative dating of those fossils are not always accurate, such as when plates collide, and it can stand the rocks up on their side, and it can change the order of the layers that way. Or you get erosion that washes away some, and then something else is laid back down on top, and things like that can change the order of the fossil layer, so that's not always reliable. So relative dating means identifying the age of fossils or rocks, comparing it to the other fossils and rocks as you look at the layers of Earth. And again, that's not always reliable due to things like floods and erosion and folding of rocks, folding of the tectonic plates. Relative dating gives scientists an approximate idea of when a fossil formed. Then we have radioisotope dating. It gives a more accurate age of fossils. Radioisotope dating measures the age of materials by comparing the amount of radioactive forms of an element contained in the material with the amount of the decaying product. Carbon-14 radioactive dating is one thing we use. All living things are made of carbon. So every living thing has a certain amount of carbon-14, which is a radioactive form of carbon. It undergoes radioactive decay, so they can look at the number of half-lives that the carbon-14 has gone through by measuring the amount that's still in there. So the half-life is the time it takes for half of those atoms to decay. Carbon-14 half-life is about 5,730 years. Okay, that's kind of a big gap there to be supposedly accurately aging things, but carbon-14 radioactive dating is used for that. The amount of carbon-14 left in a fossil tells approximately how old that fossil is. Carbon-14 is used to date fossils that are less than 50,000 years old. After that time frame, the amount of carbon-14 is so small that it cannot really be measured accurately. And then at that point, potassium-40 can be used. Potassium-40 has a half-life of about 1,300 million years. Okay. So there's going to be a big gap. Well, was it 1,300 million years ago, or was it 2,600 million years ago? So there's just that one step of a half-life there, so there's going to be a big gap in accuracy. Comparative anatomy as evidence of evolution. We have what are called homologous structures. We talked about homologous when we talked about chromosomes. That means they are similar. When we talked about homologous chromosomes, they were the same size, shape, had genes in the same locations. Well, homologous structures have a similar structure. And they look kind of the same, but their functions are different. For example, the human arm, if we look at the bones of a human arm, with the wings of a bird, with the flipper of a whale, they all have similar structures. They all have, like, we have Carpals, metacarpals, and phalanges, well, they have similar bones in the wings of a bird and in the flipper of a whale, so the structure is similar, but they're used for different things. Okay, look, the birds, of course, use their wings for flying, and whales use their flippers for swimming, so they have different functions, but they look similar. That's called homologous structures. Although the appendages are used differently, they're composed of similar bones. Then we have analogous structures. Analogous structures are body parts that have similar or same function, but not a similar structure, such as wings for flight. Bat wings are made of bones, and then we have, bird wings are also made of bones, but they have feathers on them. Bat wings are made of bones, have skin on them. And then we have insect wings that don't have bones at all. They just have thin little membranes on the insect wings. So they're used for the same thing, but their structures are different, and that is called analogous. You can make an analogy between the two, but they do not look the same. Then we have what are called vestigial structures. Vestigial structures are said to have no function in the present-day organism, but was probably useful in an ancestor. For example, the appendix has been considered a vestigial structure for a while. They would just randomly take it out just so they didn't have to deal with it later. But we're now learning that an appendix has a lot to do, so maybe some important functions in maintaining the biome of the gut. We have bacteria, and it's important that we have the right kind of bacteria growing in our gut. It helps our immune system. It helps us with nutrition. So now we're learning more about the appendix. Maybe it's not vestigial at all. Maybe it does still serve an important function. And then the tailbone. And the tailbone was just like an extension of our spine. And I'm sure it serves a purpose as balance and sitting as well. But those have been referred to as vestigial structures. Then we can look at the embryology as evidence for evolution. Embryology is the study of embryos, and again, that's the very early development of an infant. An embryo is an early stage of development in an organism. Scientists have discovered that the embryos of related organisms develop in similar ways, but if you look way back far enough in the embryo, at some point they all look kind of alike, or all pretty much alike. There's a point where human embryos look a lot like chicken embryos, and also look a lot like pig embryos, so they all, at a certain stage, most look the same. Biochemical evidence of evolution. All organisms have DNA, and DNA directs the making of proteins. Okay, so all proteins are directed by the DNA, and the DNA only has those 4 bases, or A's, T's, G's, and C's, that are arranged in certain ways to make billions of different kinds of proteins. So scientists study both the sequence and the bases in the DNA and the resulting proteins to compare organisms. Well, if we only have 20 possible amino acids to be used in all the proteins in the world, of course, they're going to be similar. The more closely related two organisms are, the more similar the nucleotides and their amino acid sequences are. And that's that. We have questions to go with those. Number 1 says features that were useful to ancestral organisms but are not useful to modern organisms. Is said to be a vestigial structure. We were just talking about the appendix and the tailbone that were thought to be vestigial but might not really be. We are learning vestigial structures. Number 2, which of these would not be used by biologists to determine the relationship between two organisms? We can look at bone structure and see if the organisms have a similar bone structure. Fossil age is another way. And DNA, that's down to that biochemical relationship that we looked at before, how closely how similar is the DNA structure, and those base arrangements. Population size. Population size is not really going to tell us much about their genetic relationship or any ancestors they had in common. It's just going to tell you what's happening in that population now. So population size will be the one that is not useful for determining relationships. Number 3, sharks and penguins both have fins to swim, but their fins developed differently. What type of structures are these? Okay, here we have similar function, but they do not look the same. Those are the ones that are analogous. They're analogous to one another, serve the same purpose, but they do not look alike. So that would be analogous structures. Number 4, whales live in the ocean. They do not have legs. However, they do have a pelvis with small leg bones attached to it. The leg bones are so small that they are not visible to the outside of the whale. What type of structures do these represent? Those would be similar to, if you look at it under an x-ray, it looks similar to a person or a dog or whatever's hip bones and leg bones. So those would be homologous structures. The appearance is the same, but the function is different. Number 5, match each statement with the phrase at the right that best describes it. Analogous structures are evidence that, analogous structures could indicate that two species face similar environmental pressures. If they are living in water and both need to swim, they would have structures for swimming. They may not be the same, but they would be analogous to each other. So they have similar environmental pressures. 6 says homologous structures. Those are the ones that look the same but do not function the same. Homologous structures are evidence that two species had a common ancestor. Number 17, vestigial structures are evidence that something that this may be evidence that one species is about to branch into two different species. They no longer need that structure, so they're fixing to change into a species that no longer has that structure. That's one line of thinking there. Number 8, embryological comparisons reveal that all vertebrate embryos look similar at early stages of development. And that is true. So A would be the best answer. All vertebrate embryos look the same at the early stages of development. When they're developing their backbone, they're all kind of curved over and look very similar at the embryo stage. So A would be our best answer. Number 9, what do scientists learn about fossils using carbon-14? That's called carbon-14 radioactive dating. So it would tell us how long ago the fossilized organism was alive. Because once they die, that carbon-14 starts breaking down. They can measure how many half-lives it has been through to get an idea of how old that fossil is. Again, that's an idea within thousands to millions of years. Not very precise. Number 10, biologists discovered 2 rabbit species living on opposite sides of a large river. So they have a geographical barrier there. They can't get to each other. What could they do to test and see if these species came from a common ancestor? Could you compare the DNA sequence? If they have similar DNA, then we can assume they came from a common ancestor. If they have the same body structures, we could assume they came from a common ancestor. Or if the protein sequences are closely related, then we can again kind of assume that they maybe came from the same group of species, and then they underwent divergent speciation and developed into 2 different species. So all of the above are correct.